Welcome to solverfy.
Hello students, in this question it's being said that assume the mating of two dogs from different breeds that both show an excessive bleeding disorder, and in one breed the condition is autosomal recessive and in other breed it is a sex-linked recessive disorder. So the dogs used or the affected male, which is a breed called as lab, and affected female, and the breed name is Doberman. So we use two breeds in which males are affected and in which females are affected. And regarding the progeny we get after breeding these two dog breeds. So in the progeny we get all normal females and all affected males. Now based on this, the question asked is which breed has sex-linked disorder and what is the genotype for that breed at both loci, because we have one of the loci is autosomal, another is the sex chromosome. So first we are supposed to find which breed has the sex-linked disorder among the two and what is the genotype of that breed at both the loci. So here the terminology I will be using is for sex chromosomes X and Y, and for the autosomal chromosome I will be writing A. So for the wild type I will add plus, and for the affected one I will add negative in case of sex chromosomes, and in case of autosome the wild type will be A and affected will be A. So now consider the affected male is suppose an sex-linked disorder. So in that case, if we consider this as an sex-linked disorder for the lab dog, which is the affected male, so in that case, we will have, the affected male dog, we will consider it, for example, it has an autosomal disorder. So in that case, sex chromosomes will be normal and it will have the autosomes will be affected, which write lowercase a shows the dog has an affected autosome. So if we consider this male dog affected as an autosomal disorder, then for the female we have to write it is the sex-linked recessive. So in that case, both the X will be affected and both the autosomes will be normal. So let's find what will be the F1 progeny. So before that, we must know what will be the gametes here. The gametes in this case will be X plus A, both two gametes, and Y, A will also be two gametes. Here, X negative A will be two number of gametes. Again, with this, X negative A, two gametes. So consider only them. So now we will consider only these four. So we'll make a Punnett square here. So this shows the Punnett square in which I put the male gametes here X plus A, Y, A, and female gametes here X negative A, X negative A. So the progeny will be X plus X negative AA, this one X plus X negative AA, X negative Y AA, X negative Y AA. So in this case, what we get, the females, because the in both the cases, whether it is sex linked disorder or autosomal disorder, in both the cases the disorder is recessive. So here one of the X is normal, so it will be a phenotypically normal female. So both the females, all females are normal. And in case of the males, also all the males here, because they have only one X, so all the males are affected. So which is inconsistent with the results we get in the question, which is stated in the question itself. The progeny we get from these two is all normal females and all affected males. So the result is consistent with the question itself, data given in the question itself. So we consider that the so here if we reverse the roles here, we'll consider the male has an X chromosome disorder and female has autosomal disorder, then we will not get the result which are consistent with the data given in the question. So by this we find that the male has an autosomal disorder and the female has a sex disorder. So the genotype of the female at both loci will be X negative, in which both the X will be affected and autosomal will be AA, autosomes will be normal. So at both the loci, the genotype of the female will be there. I hope you found the video useful. Thank you.